everyone. In this video, we're going to describe the process of protein synthesis. So in the past, we've talked a little bit about the formation of the linear chain and the fact that the linear chain can become a secondary structure, tertiary structure, and quaternary structure. And when the protein folds from its linear chain of amino acids, it becomes functional. But what we haven't done is really talk about how we acquire that linear chain in the first place. So that's what we're going to do as we sort of rewind to looking at DNA and RNA. So DNA is going to make RNA, and the RNA will be used as instructions to make the linear chain of amino acids. So I want to talk a little bit about that process and how that exactly occurs. So first, we're taking a look here at a segment of the DNA. And we know that the DNA is made of 4 different nitrogenous bases, A, C, T, and G. And those 4 bases are arranged in sequences in each of the DNA chromosomes. And it's important to understand that the arrangement of those bases, the organization of them, and the sequence of them is what provides instructions for making things inside the cells, namely proteins. So I think that it's really helpful to think of DNA as a recipe book. And in our recipe book, we'll have tons and tons and tons and tons of recipes. And we'll have to kind of imagine that it's a really big book with some really small print. And inside of each recipe, there are specific instructions in order to make whatever it is, cupcakes, pizza, anything that we're interested in making or baking in the kitchen. And I think that the next thing that we would want to keep in mind about making recipes is that we're not going to make the entire recipe book all at the same time. We're going to choose a recipe. We'll use just, essentially one at a time to make whatever product we're looking for. And in the same way, when we look at the DNA, we're not going to find that we're using all of the DNA at the same time. We're using a very tiny segment of the DNA in order to make one specific protein. And so in order to access the DNA in order to make some proteins, we actually need to unwind the DNA because when it's all tightly coiled, it's kind of hard to access it. So it'd be like trying to read the book while the book is closed. Instead, we need to physically open the book and find the page that our recipe is on. And when we do, we'll be able to read that recipe and take in that information. Now, the next thing that's important to consider is that the DNA is not able to leave the nucleus. It has to stay in the nucleus because that's where it's safe and protected. And so in order to make proteins, because we certainly don't make proteins in the nucleus, we make them in the cytosol or on the rough endoplasmic reticulum. In order to make those proteins, we need to be able to have access to the information from the DNA without actually taking the DNA from the nucleus. So what we're going to do is we're going to make a copy of just the recipe that we want. And I think that we could continue our analogy by suggesting that when you go shopping for the ingredients for your recipe, you're not going to bring the whole recipe book along with you, but instead you're going to copy the recipe or copy the ingredients from the recipe and take just the copy with you to the store in order to purchase what you actually need. So that's kind of what we're looking at initially. So our DNA segment is going to unzip. We're going to open up the DNA and we're going to find the recipe that we're looking for. And as we do, we'll be able to make a copy of just that recipe. So there are different types of RNA, and the first one we're going to consider is mRNA or messenger RNA. So messenger RNA is the physical copy of the recipe that we're looking for. Now, one thing that's important to mention is that we don't use both sides of the DNA, both sides of the ladder, in order to make one protein. Only one side codes for a protein, and the opposite side might code for a different protein, but in the context of when we want to choose our protein that we want to make, or we want to choose our recipe, we have to choose just on one side of the DNA or the other. So in the context of choosing only one side and making a copy, we're going to kind of do it in a way that's a little bit indirect. So we're going to label one side of the DNA as the template strand, and then the opposite side of the DNA is called the coding strand. So I think it's easier when we look at this in terms of letters and try to understand what exactly is happening. So the mRNA is going to, as we synthesize our mRNA, we have to remember that RNA uses the bases A, U, C, and G. So uracil replaces thymine. And as we fill in those bases, what we're essentially going to do is pair the bases to the template strand. But when we're done making the mRNA, what we're going to see is that we've essentially mirrored what we have on the coding strand. So if you look at the template strand, we're starting with TAC, and the opposite of that, because we know that T and A pair together and C and G pair together. So we see A opposite of T, we see U opposite of A because U replaces T, we see G opposite of C. But if we look then down at the coding strand in that same area, we see that A mirrors A, the U would mirror the T, and the G mirrors the G. So essentially, the mRNA is a copy of the coding strand, meaning the code or the instructions for that protein, but we match it up with the template strand because it facilitates fewer mistakes when actually making the copy with the mRNA strand. So the term transcription refers to making an mRNA template from the DNA. So when we consider protein synthesis, there's essentially two processes. The first one is transcription, which is what we've just shown, and the second one is translation, which we'll get to in a little while. Now, there's a couple more things I want to mention before we move forward. So we generally look at the DNA in terms of triplets or sequences of 3 DNA bases. And so if we look at our triplet of T, A, and C, when we make our mRNA strand, we're going to have 3 letters that are opposite of those, so A and then U and then G. And the reason I'm mentioning this a second time is because I want to label this triplet as well. So it's a triplet in the DNA bases, but the 3 letters in the mRNA are called a codon. And the reason that we bring this up is because 3 bases code for each amino acid. And so if you recall back to the original discussion of amino acids, we said that there are 20 different possible amino acids. And when we use these letters, we're essentially using them to code for each of those 20 amino acids. All right, so let's expand on this concept for just a moment. The genetic code is basically a map of all of the different possible combinations of 3 letters in a row using the bases U, A, C, and G. So if we were to look at on the left-hand side, all the way down the chart, we see U, C, A, G. That is what's going to be our first letter in each of these sequences. Across, we have U, C, A, and G again. That will be the second letter in each of these sequences. And then if you look in the actual boxes, we'll do all the possible letter combinations. So in the first box, if the first letter is U, the second letter is U, then the third letter could be U, C, A, or G. And that we'll just sort of continue that and make all the possible combinations with these letters using 3 letters at a time. The total number of possible combinations using these 3 letters is 64. So we know that there are only 20 amino acids, but if there are 64 possible combinations, that means that each of the 3 letters, we might find that more than one set of 3 letters codes for the same amino acid. And that's exactly what we're seeing here. So if you look on the top row in green in the second box, we see 4 different letter combinations. The second column of words is the actual amino acids. So, and I'm going to write them on the side as well, just so that we could see them, and you don't have to guess what they are. But we could see that there are 4 possible letter combinations, but we see only 1 amino acid, which is serine. The advantage of this, having multiple different letter combinations that code for the same amino acid, is that if there was a mistake in the copying, so we're making our mRNA sequence, but oops, we missed a letter. Well, if we got the U and the C right in this green box, then it wouldn't matter which letter U, C, A, or G we put at the end, because we still have the same amino acid that we're going to have as our outcome. So the protein will still be made correctly. So it offers some security in the sense that there will be fewer chances of mistakes. Even if there's a mistake in the mRNA, there's fewer chances of mistakes in the actual protein, and that will be considered very efficient. Now, another thing that's important to consider when we talk about the mRNA sequence is, how do we know, when you're looking at just bases and bases and bases of DNA, how do you know where to start? How does the body know where to start for a recipe for a specific protein? Well, when you read a recipe book, we know that you can tell it's the beginning of a new recipe because it starts with a title at the top, and it might start with some basic information like how long it's going to take to cook and what the ingredient list is. And so you expect a certain series of things that essentially start with a title at the beginning of a new recipe, that you know that that's the start of a recipe and not the continuation of another one. So the way that the body knows is that in the genetic code, we use AUG is the official start codon. So every time that we're starting a new protein, the first thing we're going to see is AUG. So that's kind of like the title of the recipe. And then the second question is, how do they know when to stop? How does the body know when that we've copied enough of the sequence, we've gotten everything that we need? Well, we use 3 different letter combinations that mean stop, or they're stop codons. And so when they get to those, they know that it's the end of the recipe and that we don't need to copy any more of it. And so it's really kind of interesting how the process works. I've definitely oversimplified it as far as the transcription process. There's lots and lots of enzymes that are required, and there's other steps that I've left out. But I think for the purpose of just having a general understanding of what occurs, this is kind of what we have. And now we can take a look at our list of amino acids. And I certainly don't expect anyone to memorize the amino acids. I'm just writing them because then you can see what the 3-letter combination on the right hand side of each box refers to, and you could see that it refers to each of the different amino acids. All right, now that we've finished talking about transcription, it's time to move on to the second stage. So I need to introduce the other types of RNA. So we already mentioned that we have mRNA, or messenger RNA, but we're going to need some other things as well. So the first thing we're going to need is rRNA, or ribosomal RNA. So we've mentioned ribosomes as an organelle that's responsible for protein synthesis. Ribosomes are basically made of ribosomal RNA. So there are lots of single-stranded sequences of RNA in ribosomes, and there's also some proteins. And one thing that's really interesting about ribosomes is actually that they can function as enzymes at times, and we call them ribozymes. And so the overall theme of the rRNA is it facilitates enzymatic assembly of the linear chain of amino acids. And so RNA is made of a small subunit and a large subunit, and these 2 subunits are not connected when there's no protein synthesis happening. What happens is, as the mRNA arrives to it, it'll start at the small subunit, and then the large subunit will attach itself. And then there's a little bit more of a follow-up to that, but we're gonna get to that shortly. And then what I've also labeled is there's 3 different seats inside of the ribosome. There's the A site, which is the amino acyl site, which is the point of entry for the mRNA, and then also how we're going to actually make our protein. Then we have our P site, or our peptidyl site, and then our E site, or the exit. The final type of RNA that we need to discuss is tRNA, or transfer RNA. So this drawing is basically also a linear chain of RNA, and it's folded up, and so we have some base pairing and some hydrogen bonds. And so I'm only going to draw 3 of the bases in at the bottom, but essentially this whole thing would be bases. It's not just a line of purple. So at the bottom of the tRNA, we have a set of 3 letters, and we've already labeled in DNA a set of 3 as a triplet, and in mRNA a set of 3 as a codon. In tRNA, we're going to label these letters as an anticodon. So we've already mentioned that we have our codon, it codes for a specific amino acid. So the anticodon is basically, its job is to just be opposite of the codon in terms of the letters, and that way we know that whatever letters are here, we're correctly adding the right amino acid to the tRNA. So we use an enzyme called amino acyl tRNA synthetase, and it basically adds a correct amino acid to the tRNA with a matching anticodon. So the codon that we were using in the previous screen was AUG, which codes for methionine, and so we can see that opposite of AUG is UAC for the anticodon, and then the amino acid that we're going to attach is methionine. And so that we have this enzymatic reaction that allows for the amino acid to be attached. And when the amino acid is attached to the tRNA, the correct amino acid, we label the tRNA as charged. And that basically is saying that it's ready to work. It has the amino acid. Now, there is a process of proofreading to make sure that there aren't any mistakes when the amino acid is attached. So it's, again, we're oversimplifying a little bit, but it's important to know the body tightly regulates this process to make sure that there is no errors when it comes to having the right amino acid matching the anticodon that the codon codes for. So part B of protein synthesis is translation, which is the assembly of the linear chain of amino acids from the mRNA template. In order to do the second part, translation, we're going to need tRNA, rRNA, and mRNA, so all of them. So let's take a look. Now we'll have our nucleus, and we know that the nucleus, we have nuclear pores, and the mRNA template has been made, so the mRNA can exit via the nuclear pore. We know that there's endoplasmic reticulum that's very close to the nucleus, and we can see that our initial ribosomal subunits, small and large, are not connected. So what happens is the mRNA will arrive to the small subunit, and the ribosome will assemble, meaning the large subunit will attach itself as well. Now, the mRNA does have that start codon at the beginning that tells us that this is the start of the protein, but one of the things that it will have is a signal sequence, some information contained in the initial part of the mRNA that will say whether or not the protein is going to be made for the cytosol, meaning it's going to be staying in the cytosol when it's done, and it's going to be just for the cell, or if the protein needs to be exported. So if we're using the protein just for the cytosol, then the ribosome will stay right in the cytosol and make the protein right then and there. But if we're using the protein for it needs to go as part of the membrane, or it needs to be exported from the cell via exocytosis, or it needs to be part of a lysosome, if it needs to end up in a vesicle somehow, then we need to use the endoplasmic reticulum in order to synthesize it. And at that point, what will occur is the ribosome will translocate. It will move itself over to the endoplasmic reticulum and attach itself, and then the protein synthesis will occur. So the rough endoplasmic reticulum doesn't have ribosomes attached to it permanently. Instead, the ribosomes just sort of like pop on, pop off whenever they need to, so that they can put the protein where it belongs. So first we'll take a look at the process of making a protein in the cytosol, and then we'll follow up with the modifications that occur when we make it for the endoplasmic reticulum. So since AUG is our start codon, we actually start that at the P site. Slides right into the P site. And we're going to kind of skip over a few things in terms of how that physically occurs, but we start there, but everything else, all the other amino acids will and the tRNA will sit at the A site first. So what we can see now is that we have our AUG codon, we have our UAC anticodon, which means that the amino acid has been brought over by the correct matching opposite tRNA. And then we can see that our amino acid is actually attached to the top of the tRNA, and we said that that was methionine. So this amino acid is ready to go, and then we need to bring in our next amino acid and our next tRNA in order to start the actual assembly process. So I like to think of this as very much like if you were loading and unloading at a ski lift, or if you were on a Ferris wheel, right? If you're loading on a Ferris wheel, the Ferris wheel comes down, the people get in, it moves on to the next one slides into position, you load it, and then it just sort of continues. So the same thing is going to happen here. So our next amino acid is attached to the tRNA is going to slide into place, and then we will have a chemical reaction that will occur between those two. So we now have a covalent bond or a peptide bond between those two amino acids. And then as the process continues, the initial tRNA that's in the P site will move to the E site. The tRNA that's in the A site will move to the P site, and a new amino acid will slide in to the A site, and we'll again have this chemical reaction between the green amino acid, which is proline, and the next amino acid that's coming in. So I didn't initially draw the mRNA to be long enough, but we'd have to imagine it would be, a very lengthy strand of mRNA in order to make a protein. So I want to fast forward a bit, and so if we were to do sort of like a time lapse, we could see that this is what has happened. And now we can see that our linear chain of amino acids is extending out of the ribosome, and we can also see that we have 3 tRNAs, one in the E site, one in the P site, one in the A site, and then we'll have our next tRNA that's kind of waiting, and they'll all sort of move in together, and they'll all sort of just move along in succession. So this process just keeps going until we've made the entire linear chain that we're looking for to complete the protein. And in the cytosol, there will be some enzymes that will help to fold up this amino acid into its secondary, tertiary, and possibly quaternary structures. All right, so now let's backtrack for a second and take a look at what happens if the mRNA contains a signal sequence that says that the protein needs to be made at the endoplasmic reticulum because it's going to be exported from the cell or placed into the cell membrane. So the ribosome will attach to, there are several receptors that are present on the endoplasmic reticulum, and it'll attach to it. And so what essentially happens is as we make our linear chain, instead of feeding the linear chain into the cytosol, we're literally feeding the linear chain through a tunnel or a channel that is in the endoplasmic reticulum, because don't forget, this is phospholipid bilayer, this whole blue line here. So we're basically moving that linear chain into the lumen of the endoplasmic reticulum. And as we do that, now it's contained inside of a liquid. It's not the cytosol, but it's contained inside of a liquid. And we'll be able to fold up that protein in there. There'll be enzymes inside that we can use. And then when we need to move it to its next destination, it'll pinch off in a vesicle from the endoplasmic reticulum. It'll go to the Golgi, where it can be receiving its shipping label. It could have some additional processing occurring, and then the options for it after that include fusion with a lysosome so that we can increase the number of lysosomal proteins, or it could be secreted. Now, I do want to mention that secretion can be constitutive or regulated. So constitutive secretion basically means that as soon as the protein is ready, it gets dumped out, and that just kind of happens on a regular, recurring basis. Regulated secretion means that there has to be some sort of a stimulus in order for the actual exocytosis process to occur. So we might need some type of hormonal stimulus, or we might need some type of something binds to a receptor, or there's some type of cellular communication occurring. Something has to physically happen that will trigger the cell to actually perform the exocytosis of those proteins. So it's really very interesting because a cell does have the ability to do either one, to just produce a mass amount of proteins and just dump them out there when they're ready, or hold onto them in little granules or vesicles until the time is right, and then release them in a burst. Now, the final thing I wanted to mention in the context of making proteins at the endoplasmic reticulum is if we wanted to make a protein that is for the cell membrane, we actually are going to attach to the ribosome to the endoplasmic reticulum again, and then we're going to make our linear chain of amino acids. But what's really interesting about this process is that in a protein, some parts of the protein are going to be on the inside of the cell membrane, and some parts are going to be on the outside, and some parts are going to be right in the middle, like right as part of the cell membrane. So it's kind of like an interesting stitching process that occurs. So the linear chain will be fed into the inside of the cell, and then when it gets to a part that needs to be part of the cell membrane, and usually it will mirror the characteristics of the membrane, so instead of being hydrophilic, it'll be hydrophobic, then those amino acids will be just kind of linear, placed in a linear chain right in the membrane. And then if there's an outside part, we'll make the outside part will be fed outside of the endoplasmic reticulum, and then if we need to go back to the membrane again, we'll do that. And so I know it's kind of tough to tell in this drawing, but our time-lapse version shows us we have some of the green is actually in line with the blue, meaning that it's right in the membrane, some of it's on the inside, some of it's on the outside. And then we make our vesicle to transport this to the membrane. What we can see is that we have membrane proteins that are actually physically part of, not inside the vesicle, but physically part of the vesicle boundary. Which is also phospholipid bilayer. And then we can travel that to the cell membrane, and it gets just placed into the membrane, and now, boom, we have all these extra proteins that we didn't have before, or maybe we're replacing some that were broken down. So that concludes our explanation of protein synthesis. Hope you enjoyed it. Thanks for watching.